Hello and Assalamualaikum. We are from Group 1A and this is the poster for our project entitled Adsorption Isotherm and Kinetics of Congo by Biowaste Adsorbents in Textile Industrial Wastewater Treatment. Here is the list of our group members. As an introduction, textile effluents are known as toxic for containing many metal complex and reactive dyes. In this study, the adsorbate conglomerate dye itself is known as a carcinogen. Therefore, the adsorption by biowaste adsorbents is chosen due to its low cost, simplicity, and flexibility in its operation. For the market analysis, the water treatment technology market is expected to grow over $176 billion by 2025 all over the world. This is because the available clean water sources can no longer support the growing populations due to the increasing pollutions by various industries, and this is where the treated wastewater is needed to solve the problem. Here are the three objectives of our study.
Units for the wastewater treatment plant. Firstly, effluents from several pre-treatments and dyeing processes are transferred to the equalization basin. Then a set of impellers and aeration system work together to mix the wastewater in the basin. From the equalization tank, the homogenized wastewater is transferred to the fast stirring tanks where the coagulants such as lime and ferrous sulfate are introduced. The wastewater is then transferred to a larger tank by overflow where the coagulants and flocculation takes place. Wastewater solids are then separated from the aqueous phase and then dried in drying belts. Before final treatment step, hydrochloric acid solution is introduced for neutralization and a series of sand pressure filters remove the suspended solids. Finally, biowastes are being used as adsorbents to remove the red Congo dyes and heavy metals. From there, treated wastewater can be safely released into nearby ponds or streams without any harm to the environment.
The first unit we have from the wastewater treatment plant is the equalization basin, used to maintain consistent influent water flow by retaining high flow fluctuations. Next, impellers and aeration system, where air is pumped into the tank, which then adds oxygen to the water and promotes microbial growth in the wastewater. Thirdly, fast stirring tanks, the most widely used type of reactor in the process industry. They contain one or more impellers mounted on baffles or draft tubes. And then we have coagulation flocculation tank, which is to enhance the ability of the treatment process to remove particles. Number 5 is sand pressure filters to remove suspended matters, floating, and sinkable particles. Inside, the wastewater flows vertically through a fine bed of sand, and particles are removed by way of absorption or physical encapsulation. Last but not least, biowaste material absorbance tank. Absorption of dyes by biowaste is a novel approach, and it's preferred compared to activated carbon due to its high surface area, porous structure, high absorption capacity, and lower cost.
As observed in the left figure, the equilibrium time for GNC is 80, 60, and 30 minutes, and for EC is 90, 60, and 40 minutes at 308, 313, and 318 Kelvin respectively. Both adsorbents reach equilibrium adsorption faster at higher temperature. At higher temperature, the dye ions gain higher mobility, thus increasing the chances for adsorption. As observed on the right figure, the removal efficiency of GNC decreased from 94% to 73% from pH 2 to 12. For EC adsorbent, the removal efficiency decreased from 75% to 51%. This phenomenon is because at low pH 2, there is a significantly high electrostatic attraction between the anion dye and the surface of adsorbent that is positively charged. The lower absorption at high pH level can be explained by the excess presence of hydroxide ions. They will compete with dye anions for adsorption sites. On the left figure, it is observed that the higher the adsorbent dose, the greater the removal efficiency up until a certain limit. Increasing adsorbent dose increases the adsorption rate, because more surface area are available for adsorbent to adhere on the active sites. On the right figure, it is observed that for both GNC and EC adsorbents, that as the initial concentration of dye increase, the equilibrium adsorption capacity increases too. More adsorbate molecules are available to be absorbed, so the actual amount of dye that is absorbed on adsorbent surface rises too. Meanwhile, the removal efficiency decreases as the initial concentration of dye increase. The active sites of the absorbent surface become saturated, hence slowing down the adsorption and ultimately reducing the removal efficiency.
Hello ladies and gentlemen, I am Harihaaran Murthy, and today for my part I am going to talk about and determine the best fit of the adsorption isotherm. Because there is insufficient data in the removal of Congo rate experiment, we had to use another similar experiment, which is the removal of methylene blue from textile wastewater. In order to decide whether the adsorbent surface is homogeneous or heterogeneous, the Langmuir and the Freundlich models were used, and the data were used to sketch these two graphs. On the left hand side, it is the Langmuir adsorption isotherm, in which CE over QE against CE is plotted. From this graph, the coefficient of determination, which is the R square value, is equal to 0.99, which is very close to 1. On the right hand side, as you can see, it is the Freundlich adsorption isotherm, in which log QE against log CE is plotted. And the coefficient of determination, the R square value, is equal to 0.98. Although both have R square values close to 1, the one in the Langmuir adsorption isotherm is higher, and therefore we can conclude that the Langmuir model best depicts the adsorption component in this experiment, and the adsorption interaction is a homogeneous and single layer one. That is all from me today. Thank you very much.
As a conclusion, we are distributing the results and discussion into three parts to satisfy those three objectives. As for the part A, we conclude that different conditions will highly affect the adsorption and the removal efficiency. Thus, we can determine the optimum condition from the experiment. For part B, for both methylene blue and GNC, it is the pseudo second order for both process. And part C, we can determine that Freundlich isotherm describe the best for the process of the adsorption. So for the safety and other consideration, the first one, we need to consider the human error, since it involves lots of measurement and data collection and also calculation. The second one is about the stirring process for the methylene blue in order to keep it at a constant rate for each process. That's all from us. Thank you.